Now that you know how some things move across a membrane, things like ions and macromolecules and proteins and nutrients and waste, today we're going to look specifically at how water and only water moves across the membrane. Okay. And this is called osmosis. Osmosis is when water moves from an area of high concentration to an area of low concentration through a semi-permeable membrane. All right. So in this diagram here, the blue circles are water, the red is some kind of solute, like let's say starch or sugar or salt. It doesn't really matter. But the idea here is that that solute is not moving, right? It must be something that can't cross the membrane right now. And so the only thing that's going to move is water. So if we look, where is there more water? Well, just visually, there's more blue dots on the left side than the right side. So the water would move from this left side here across the membrane to the right side. Okay, that's all osmosis is talking about. If you've taken human A&P, or if you remember back to freshman bio, these are words that you've probably heard before, but there are 3 different environments that cells can be in: hypertonic, hypotonic, and isotonic. Okay, we're going to go through each one of those and refresh your memories on those. Hypertonic, think hyper meaning lots, like lots of sugar, for example. Okay. And so what we're doing is we're comparing the cell to its surroundings. So here's an example of a cell that's in a hypertonic solution. So the blue dots are the water, the red dots are the solute. And so notice that, again, with osmosis, we're really focusing on water and water movement. Okay, but let's just take a look. Notice that outside the cell, there's a whole bunch of red dots and some blue dots. But inside the cell, there are just a couple of red dots and a whole bunch of blue dots, right? So outside the cell is more solute and less water, and inside the cell is less solute and more water. So remember, water is going to move from high to low, and in this case, high is inside the cell, low is outside the cell. So water would move from inside of the cell to outside the cell. Water is going to leave our cell here, okay? And so this is going to cause our cell to get all shriveled up. Next up, we've got hypotonic. So this is kind of the opposite of what we just talked about. Notice here, we've got a lot of blue water outside the cell and just a little bit of solute. Look at inside our cell. We've got a whole lot of red solute and less blue. So our area of high concentration is going to be outside of our cell. Our area of low concentration is going to be inside of our cell. So which way is water going to move? It's going to move into the cell. And because water is moving into the cell, it's going to cause it to swell and maybe possibly burst. Lastly, we've got isotonic. Okay, so this is where things are going to be equal concentrations. We're not going to take the time to count here. We have equal blue dots and red dots on the outside as we have on the inside. So which way will water move? Nowhere really. Now, that doesn't mean that some water might not move. Okay, there might be a water molecule or 2 that comes in, but then 1 or 2 will go out. So overall, the net change, the overall change is that there is no change. Okay, so our cell is going to stay the exact same size and not swell up and not shrink down. So here is a summary. Okay, and one thing to point out here is notice the difference between the animal cells up top and the plant cells down below. Pretty different, okay? So up here with the animal cells, these are red blood cells, but it's true for any of our cells. Notice what quote unquote normal is for an animal cell. That would be isotonic, right? So an isotonic solution means our cells just kind of aren't going to be, just going to be kind of in this same state here, right? It'd be about the same as their surroundings. If we were to possibly drink too much water, and let me tell you, it would take an absurd amount of water, but it's possible. If you were to drink enough water, you could create a hypotonic solution outside of your cells, and water would go into your cells and they could possibly swell and burst, which would not be very good. Okay. And if you were to drink a hypertonic solution, like let's say if you were stranded at the ocean and you decided to drink ocean water, well, what would happen? Water would leave our cells and they would shrivel up and you'd probably die, at least eventually, okay? Now, let's go down to this bottom row here with the plants. Where's normal? Oh, well, it's not here in isotonic. It's over here. Oh, interesting. So with plants, we need our cell membranes to be nice and expanded and to be pushing against our cell walls because that's what keeps our plant cells upright. That's how plants can stand without bones, okay? And so for a plant cell, its happy place is actually in a hypotonic solution when it's got water actively moving into it. So it's nice and expanded and it's kind of swollen. Plants, if they're a little bit underwater, we notice they might start being a little bit droopy. That's going to be in an isotonic solution. And then plants in a hypertonic solution, so much water is going to leave that they're going to shrivel up and probably die. Okay. So there's our difference there between hypertonic, hypotonic, and isotonic, as well as the differences between plant and animal cells. Okay, so those basic concepts are what you need to know for this lecture.